Hey guys, welcome back to One Stop Biology. So in today's video, we are going to discuss all about nucleus. So guys, nucleus as a cell organelle was first, discovered by Robert Brown, and it was discovered in 1831, right? So basically, later on, what Fleming, another scientist, did was he stained nucleus with basic dyes, and he basically came up with the name chromatin. Right, so nucleus was first seen as an organelle by Robert Brown, but the chromatin part of it was discovered by Fleming. Now, the interphase nucleus, that is when the nucleus is not dividing, right? So, in the cell, when the nucleus is not dividing, it is known as interphase nucleus. Interphase nucleus. So the interface nucleus has, highly extended and elaborate nucleoprotein fibers, and these fibers are known as chromatin. These are chromatin fibers, basically, right? So they have these extended nucleoprotein fibers, which are chromatin fibers. Then they have nuclear matrix, and they have some spherical bodies. These spherical bodies are known as nucleolus or nucleoli, plural. Right. So when you see the electron microscopy, you'll see that there is a nuclear envelope. So now this nuclear envelope basically consists of two parallel membrane with the spaces. Right. So basically they say if you see a nucleus, there is a nuclear envelope which has two membrane. Right. Now, there is a space between these membrane, which is known as perinuclear space. It is known as perinuclear space. Now, this basically space is 10 to 50 nanometer, and this basically forms a barrier between the material which is present inside the nucleus and the material which is present in the cytoplasm, which is outside of the nucleus. So basically, the outer membrane, this membrane, the outer membrane, this membrane usually con- basically remains continuous with the endoplasmic reticulum, and it also bears ribosome. So if you remember, we studied that you have endoplasmic reticulum outside, right? And these endoplasmic reticulum bear ribosomes, and these are sites of protein synthesis, right? So basically the DNA and RNA is materials present inside, and it is transferred outside into the cytoplasm for translation for protein synthesis, right? So the outer membrane forms the endoplasmic reticulum in continuation, which has ribosomes. Now, there are number of places on the nuclear envelope which is basically interrupted by minute pores. So there are places in the envelope which has pores, right? Which has these perforations, basically, which has pores, and these are formed by the fusion of two membranes. So it basically, these pores are known as nuclear pore. Right. So these pores are passages through which the movement of RNA and protein molecules happens in both directions, right, between the nucleus and the cytoplasm, right? So normally there is only one nucleus per cell, but there might be some variations as well, wherein you can have multiple nucleus. And there are also some cells which lack nucleus, like erythrocytes of many mammals. They lack nucleus, right? Now let's come to the nuclear matrix first. What exactly nuclear matrix contains, right? Now the nuclear matrix is known as nucleoplasm. This matrix contains nucleolus and the chromatin fibers, right? So the nuclear matrix is also known as nucleoplasm. Like we have cytoplasm in the inside the cell, we have nucleoplasm inside the nucleus. Now this contains one nucleolus and second chromatin fibers. Right. So let's see what exactly nucleolus is. The plural is nucleoli, guys. Now, this nucleolus are nothing but spherical structures which are present in the nucleoplasm. Now, if we talk about the content of nucleolus, the content of nucleolus is continuous with the rest of the nucleoplasm. It is just that it is not a membrane bound structure. Right. So it has nothing but the same content as nucleoplasm has. Right. It's just that it is not a membrane bound structure, and it is site for ribosomal active ribosomal RNA rRNA synthesis. Remember that it is site for active rRNA synthesis, right? Now there are various larger and numerous nucleolus present in cell which actively carry out protein synthesis as well. So they carry out protein synthesis as well, right? So guys, if you see here the structure of nucleus, if you see this structure, this is the structure of nucleus, where inside these fibers are chromatin, you have nucleoplasm, fluid. See, these are the pores. So these are nuclear pore. You have nucleolus. The outside, this is nuclear envelope. These are ribosomes. And this is endoplasmic reticulum. Right now we are talking about this nucleus, right? So this nucleus has basically this nucleolus is very similar to nucleoplasm, and it has basically this nucleoplasm has nucleolus and chromatin fibers. Now these nucleolus are site of rRNA, ribosomal RNA synthesis, and they carry out protein synthesis as well, right? Now we said that the interphase nucleus has chromatin, right? So basically these chromatins during different stages of cell division, they form structural chromosomes. They form chromosomes, these chromatin, right? They form chromosomes in different stages of cell division, right? So in place of nucleus, you'll find chromosomes. Now this chromatin contains what? Chromatin contains DNA and some proteins which are histone proteins, right? So they contain DNA and histones or histone proteins, right? Now they also have some non-histone protein and RNA as well, right? So they have DNA, this plus some non-histone protein and RNA as well, right? So if we talk about a single human cell, guys, a single human cell has almost 2 meter long thread of DNA which is distributed among its 46, that is 23 pair of chromosomes, right? So a single human cell has around 2 meters long DNA, thread of DNA, right? Now every chromosome that we see, every chromosome has a primary constriction, They have a primary constriction, right? And these are known as centromere. These are known as centromere. So if we talk about the chromosomes, right, you'll see that every chromosome has a primary constriction or centromere, and on this centromere, on this side of centromere, there is a disc-shaped structure which are known as kinetochores, right? So if you see this structure, you will see that this basically is a chromosome. This is a chromosome. Now these are a pair of sister chromatid. So these are a pair of sister chromatid. Now here, this center is centromere. There is a construction which is centromere, and these two structures that you see are kinetochore. So basically, this is one chromatid, and this is one chromatid, right? So now, based on the position of the centromere, there are different types of chromosomes. So there are four types of chromosomes on the basis of the positioning of this centromere, right? So let's see what are these different types of chromosomes, right? So the first one is metacentric. By metacentric, we mean that the chromosome has middle centromere, which forms two equal arms of the chromosome. So metacentric is nothing but having two equal arms of chromosomes, right? So the first one that you see here, this one, this one, this one is metacentric, right? So you have basically centromere. These are nothing but satellite. These are satellite, and this is known as secondary constriction, right? Now the second one is submetacentric. Now submetacentric, what happens here, that the centromere is slightly away from the middle, and that results in one short arm and the other long arms. The one you see here, this one is submetacentric. Now here what is happening is, this is the short arm and this one is the long arm, right? So basically, if you see here, it's exactly at the center. Here it has shifted towards one of the ends, and that is why one arm of this chromatid or the chromosome is shorter and the other one is longer. Now the third type is acrocentric. Now acrocentric, what happens is that centromere, now this constriction now moves more towards one of the end, and that what happens is that one of the arm is very short, right? Extremely short arm, and the other one is very long, right? So this is the centromere, right? And the last one is telocentric. Now telocentric means terminal centromere. So in telocentric, what happens, it shifts even more towards the shorter arm, and it's almost at the terminal. So here you see the short arm is so short, teeny tiny, right? So we have four different types of chromosomes depending upon the position of centromere. The first one is metacentric, which means centromere in the middle and two equal arms. Second one is submetacentric. So it has now shifted towards one of the ends, and now the one of the arms are short and the other one is long. Then we have acrocentric. So it moves even more towards the end, and one of the arm is now more shorter, extremely shorter, and the other one is very long. And the last one is telocentric, which means terminal centromere. So it has now shifted towards one of the terminal, right? So these are four different types of chromosomes. Now, sometimes, the few chromosomes have non-staining secondary constriction as well. So this is secondary constriction, guys. This is secondary constriction, right? And these constrictions are at a constant location basically. So this gives the appearance of a small fragment, and these fragments are known as satellite. These are known as satellite, right? So this is all about nucleus, guys, right? I hope we have finished the entire basics that is given in the NCERT as per the class 11 syllabus. I hope you understood this video very well. So do not forget to like it. Please share it with your friends. And if you're new to the channel, please do not forget to subscribe to the channel and press the bell icon. And guys, if you have any doubt in this topic, in this particular topic, please do not hesitate in asking doubts. You can either ping me over WhatsApp, the number is given in the description of the video, or you can also ask your doubts through commenting. Just comment on this video and I'll revert to your questions. But do not forget to give comments and let me know how did you like the video. Thanks for watching the video, guys. See you again. Bye bye.